Hello and welcome to BIOS Frontier Science. We're thrilled to welcome Lili Yang, University Professor of Microbiology, Immunology, and Molecular Genetics at UCLA.
Thank you for having me today.
Thank you once again, Lili, for joining us. To kick things off, Lili, can you share a brief intro with us?
Sure, yeah. I was born in a family of engineers. Both of my parents are electrical engineers, so I guess that have imprinted the engineering instinct in my genes. I became fascinated about biology when I was in middle school and became especially attracted by the subject of immunology when I was in college. This motivated me to pursue a PhD in immunology at Caltech, studying with David Baltimore, a Nobel Laureate recognized for his many groundbreaking discoveries in modern molecular biology and immunology. During my PhD study, around the year 2000, cancer immunotherapy achieved several remarkable breakthroughs, including the success of the immune checkpoint blockade therapy and the CAR-T cell therapy. I saw a perfect combination of my engineering instinct and immunology interest and decided to develop my research career in the field of cancer immunotherapy with a focus on gene and cell engineered cancer immunotherapy.
That's fantastic. And before diving deep into specific projects here, one question we really like to ask our guests. Throughout your career, Lili, what has been your North Star, the common thread, if you will, that ties your work together?
Yeah, that's a very good question. I think it must be revolving around engineering and biology, and now combined in the term of engineering immunity. It's the name I gave to my research laboratory. And the meaning is that we're trying to empower our own defense system, the immune system, to fight off the diseases using the genetic tools and the cell therapy tools.
Thank you again, Lili. I'm going to pass it over here to my co-host Minju to take over our first section here.
Hi, Manju.
Great. Hi, great. Thank you, Drew. And thank you, Lili, again for joining us. So diving a little bit into your research, your lab focuses on deciphering the cellular and molecular mechanisms of controlling anti-tumor immunity to create the next generation of cancer immunotherapies. And in particular, your research has recently focused on your efforts in identifying new checkpoint inhibitor molecules that can serve as potential drug targets. So first of all, can you provide an overview of your approach to uncovering these molecular mechanisms of immune surveillance?
Sure, yeah. It comes from some very basic idea. The idea is during the immune cell fighting of the tumor cells, if some molecular mechanism is particularly important, when you compare the behavior of the immune cells prior to encountering tumor and after encountering tumor, there must be some changes, either upregulation or downregulation. So that is the base for our screening strategy. So we compare the genomic profiling, behavior of the cells prior to the encounter tumor and after the encounter tumor, and search for the changes. And if there's some changes coming out, popped out, it's new, haven't been studied, but has indication about linking to the anti-tumor immune reaction, that we pick those molecules and follow through and studying this.
Great. So how does your research on these new checkpoint inhibitors or molecules contribute to our current state of understanding with immunotherapies like PD-L1, CTLA-4 targets in cancer landscapes right now?
Yeah, so the beauty of the biological system, including the immune system, is that there are multiple control points, right? During the fight against the tumor, there are points to control the recognition of the tumor cell by immune cells. That's point to control the infiltration of the cells into the tumor. And that's point to control the function of those cells at the tumor site. Okay, and there's a group of molecules involved in this controlling network. Okay, some works at this particular point, another works at those particular point. I think the success of the PD-1 and the CTLA-4 blockade story is very encouraging, because that tells us if you pick the right molecule and the right checkpoint and interfere with those, will make a significant impact on the overall anti-tumor response. And of course, by no means PD-1 and CTLA-4 should be the only two magical molecules that we can target, right? That's, again, the group of them. But how important each molecule is, and probably we're tied to a particular disease, like anti-PD-1 treatment work for many types of cancer, but not for all cancers, right? And for each type of cancer, it works for some patient, not all patient. So that means the other molecular the regulatory network must play a role over there. And by looking, for the new, molecules, expanding the family of those target, that we may, have a better chance to target more cancer and treat more patients in the future.
Yeah, that's really interesting. And I particularly love like the translational patient-focused theme that your research seems to have. So I know you talked a little bit about this in your intro, but can you share what pursued you to focus especially on tumor immunology, and in particular, understanding the landscape of the interactions between immune cells and the tumor microenvironment?
Yeah, so as mentioned, the multiple, things need to happen for immune cell to successfully fight off tumor, right? They need to find the tumor, get into the tumor site, and remain functional at the tumor site. So the players are more than just the immune cell and the tumor site. There's the entire microenvironment at the tumor site get involved, right? So like to make sense happen, we need to take into consideration of the participation of all the major players over there. Turns out that the tumor microenvironment, people get more and more realized that it really significantly impacts the whole outcome, right? There's positive regulation, that's mainly negative, actually, regulation of the immune reaction over there. So devise some immunotherapy to help the immune cells themselves, plus, help modulate the tumor microenvironment, probably are the key, to really make immunotherapy successful.
Yeah, that's really interesting. And now jumping a little bit more into the engineering side of your work, you are also super widely recognized for your work on hematopoietic stem cells, engineered into natural killer T cells. So iNKT cells in adoptive therapy, which is pretty impactful in the field of advancing cancer immunotherapies. And you have shown that both in vitro and in vivo, pretty powerful work on the abilities of these natural killer T cells to induce anti-tumor immunity. So a little bit on this front, can you expand on the development of these HSC natural killer T cells and what you're doing specifically to engineer or enhance these certain properties?
Certainly, yeah. So let's consider a drop of the blood, okay? It contains about 10 million of the cells. Within those, we have a small portion are called white blood cells, and those are the immune cells. A very small portion of that immune cells are the NKT cells that I'm studying, okay? We have around like 10 NKT cells in one drop of blood, a very small number, okay? But why we are interested in studying them? Because those cells are very powerful tumor fighters, okay? So although they are smaller number, but they respond stronger and faster than the regular lymphocytes like T cells. The greatest limitation we already touched is that small number. Okay, so how to increase the number, get more, because in the fight against the tumor, number also matters. Immune cell, as powerful as it is, may be fighting off 10 tumor cells, but ask one cell to fight off 100,000 of tumor cells. They need a group of themselves, to do the work. Okay, so the idea behind the HSC, iNKT cell technology is that we try to create a way to overcome this critical limitation. Okay, so like we take advantage of the stem cell engineering tool. We go back to the ultimate progenitor cell, which gave rise to all the lineage of the blood cell, including immune cell and as well as NKT cells. So the name of that called blood stem cell, another name called hematopoietic stem cell, shortened as HSC. Those cells live in the bone marrow of adult human, also exist in the cord blood, actually in a much enriched abundance, okay? And cord blood resource of the HSC can be a really good source, to produce, to engineer and guide them to differentiate into the NKT cells. So my lab is currently focusing on, produce some manufacturing protocol, really makes this happen.
Yeah, that's so innovative, the way that you're doing this. So as you've described, a large part of your research is bridging those roads from basic to translational research, and then bringing those functional discoveries into the clinic. So for this, can you share a little bit more about the intersection of the mechanistic approaches to immune surveillance and how you're using that to engineer immunity in cells for clinical development specifically?
Certainly, yeah. As mentioned, like we need to really go down to the basic, to how the anti-tumor immune reaction happened and why sometimes it's successful, why sometimes it's not successful, okay? So the different, our scientists do is to break down a question into the pieces like we can attack, right? So like the tumor recognition, for instance, how to make the therapeutical cells recognize tumor better, right? Very good example is the CAR engineering technology, the chimeric antigen receptor, right? So it's a synthetic molecule. It's not existing at all in nature, but it smartly combined the recognition ability of antibody and the signaling part like trigger a T cell to respond. So something like this can be certainly adapted, into the cell platform we're working with, right? So we're working with this NKT. They already have their own T cell receptor that can allow them to attack tumor at a certain level, but we can certainly give them more, right? Give them a more targeting molecules. They give them a CAR, direct them better and recognize and attack the tumor better. Another is the trafficking, right? So we want the cells to be able to very actively find the tumor and go to the tumor site and infiltrate into the tumor site. And there's a group of molecules doing that. Our cells already have some, but we can give them more, right? So especially for specific type of cancer, we know like what are the special addressing trafficking molecules will attract the cell better to that particular site. And we can enhance the cell for that part. And yet another part is the microenvironment we talk about. Once the cells get into the microenvironment, how they stay active, not get tired and get exhausted, and eventually, loss of function at the end. That's yet another group of molecules, controlling the function, controlling the memory and the persistence of those cells, which we can enhance on our cell product. So all of those aspects, the power of the gene engineering, cell engineering, technology nowadays it's possible, to put all of those bits together. And likely, probably more than one engineering, strategy is necessary to make really the perfect product at the end.
Yeah, it's always so fascinating to learn how brilliant scientists like you are able to have such a solid understanding of the biology and translate that into a clinical application. So in your opinion, what are some of the crucial aspects to consider when designing the translationability of these cell models?
Sure, yeah. So that is something both overlap and a little bit different from the basic research side. From the basic research side, we can fascinating any possibilities, right? We can think about many smart strategies. We can have 10 or more than that, a kind of engineering steps incorporated to make really the idea product. But in terms of translation, I think the sort need to be a little bit adjusted. To make it really translatable, a product need to be robust, okay? It need to be repetitively can be made at a larger scale and can be really a kind of robustly delivered. And the second is it should stay as simple and practical as possible, right? So taking the example, I can have 10 different engineering steps to make a product. I may only need 2 or 1 engineering step to make a product 90% as powerful as the one, like I described previously. Which one have the better chance or really make the real impact to translate, get into clinical, maybe commercial in the future? May not be the first one, maybe the second one. But those are, of course, a decision needed to be made and very carefully assessed for the research part to say, like, if lost that 10% of that function can really gain me the robustness and the really practical way of manufacture the cell and deliver and cover more cancer and more cancer patients, maybe that's a price we're willing to pay.
Yeah, great. That was such a great overview of your research. So now handing it back to Drew.
Of course, and while we're speaking on translation, wanted to ask Lili specifically about your clinical research. One quick addition here is, I mean, in addition to your basic and translational research in the lab, your study has led to 2 clinical trials with more upcoming. I would just love to take a pause here, and can you just give us an intro to the clinical trials and what aspect of your lab is being translated into patient populations currently?
Sure, yeah. So let me start with the clinical trials already happened, right? And then I'll touch base on the upcoming clinical trials. Right, the trials already happened. There are 2 clinical trials. One is done from the academic side. One is done from the industry side. The academic side, I'm working with my clinical, collaborators at UCLA hospital right here on campus next to me. Just walk down 10 minutes, I can find all my collaborators over there. It's a clinical team, led by Dr. Antonio Ribas and many, members in his clinical team. So that idea is to translate one, research initially developed in my lab. We find those that tackle the first part of the tumor, targeting is to recognize tumor. So we isolated one molecule called the T-cell receptor, recognize common tumor antigen called NY-ESO1. Okay, so this, and we equip the immune cells, in particular T cells, with that T cell receptor, and then give back to the patient. Okay, the idea is that now with this particular, molecule we give to the immune cells now can better recognize tumor. So that's the take care of the first step of an anti-tumor response. Yeah. And the second clinical trial run from industry side, it's more on the cancer vaccine, side. It's a technology developed in the lab, and then it's licensed by the company, and then they run the clinical trial. Actually, it goes through all the way to phase 3. So the idea is to more like stimulating, not really giving a recognition molecule, to the immune cells. Is to, in a vaccine platform, like we packed all the necessary information to train the immune system, recognize a particular tumor. We gave an individual that training information in the context of a lentivector encoding that particular tumor recognition, antigen, everything. So the overall consequence will be, by injected with, this training material, that individual patient can really engage their own immune cells and stimulate the part of the immune cell have pre-existing ability to recognize tumor. Now give them a boost them to grow more, to expand and give them a better function, to fight tumor. So those are the 2, already happened in the clinical trial. The upcoming clinical trial, we have like a pipeline of things lined up we want to do, which is at the different stages of the translation, and the spanning the quite large category of blood cancer and solid tumors. And that's involve effort from the academic side, which likely many things will happen at the UCLA hospital, and the industry side, the technology licensed by RP Bio, like they are developing it from the industry side. It's more about the cell therapy, and we want to convert the very successful CAR-T cell therapy now off the shelf in our own special format of CAR-NKT cells.
Of course, it's amazing to see the blend between academia, industry here to push this trial forward, and just super excited to see the progress going forward, and looking forward. We'll be on the sidelines cheering you on. And so as we just continue, we just wanted to continue the conversation. Would love to gauge your thoughts. I mean, as the field continues to advance in CARs and NKT cells, in immunotherapies, what are some of the technologies you are most excited by?
Yeah, so the technologies I will be most excited for converting autologous cell therapy into off-the-shelf cell therapy. I think that technology will likely to be one of the most exciting advancement in the coming decades. For instance, as much as we love CAR-T cell therapy, I mean, it's amazing. It's like a revolutionary, right? So it really saves patients, but only a small portion of a patient, right? Like from the disease. Treating patient like one by one, it's really put a big hurdle on making the therapy accessible, to many patients and to many different diseases. Okay, so when we talk about medicine, we think about you can walk into a store, right? Your doctor diagnose something that can just prescribe and something can be delivered. Pick it up at the hospital and be given. That is what we think about medicine, and that is traditional medicine is. Can be small molecule, can be protein, based drugs, but something ready to go off the shelf, right? So the cell therapy at now, like you need to hospitalize a patient. Not everybody can, get into a facility, really being able to coordinate all of those events to happen. Blood needed to be drawn, and there's a period of time needed to be waited, to make the cell product. It may be successful, it may be unsuccessful. And meanwhile, at the time of the waiting, the disease situation for a patient may change, right? It may go, beyond the stage, like can be best, to receive the therapy. And then after all of those efforts, the cell product made can only be used to treat that single patient. Okay, it cannot be generalized and given to the other patient. It's because the special way, something called the MHC, HLA matching, thing need to take into place, when you transfer cells between people. So all of this just put a very, a kind of a bigger hurdle over there for the application. So making cell therapy into a form of our traditional medicine, like really off the shelf, being able to pre-made and categorized, and the doctor can just prescribe, kind of when a patient diagnosed, and then the patient can receive the treatment immediately without waiting. And that also can greatly reduce the requirement for, like it can be, many hospitals now may be able to do it, compared to just a very limited, big medical center can really hospitalize patient and manufacture the personalized cell product.
Of course, and I appreciate you sharing your thoughts on the current challenges of the industry. And as we look towards the future, I'd be generally curious about your thoughts within the next decade. How do you really see the field from your own perspective of cancer immunotherapies changing over, say, the next 10 years with this technology?
That's a very good question and exciting question. Think about the immunotherapy. The cancer immunotherapy, see, it's really booming in the past 1.5 decades, no more than that. Well, people tried, the generations of scientists and the clinicians tried very hard, far beyond, before that, but it really started to work. It's in the past 1.5 decades. You're asking me what will happen in the next decades? My guess, expectation, would be the booming will continue to happen, especially now we have the newer technology, kind of a better, kind of resources pulling into the field because of the success we already saw. If I want to really narrow down on a few, a kind of highlighted points, I would say number one, we likely will see more choices for the checkpoint blockade therapy. Okay, we may have another magic one, as powerful as anti-PD-1, maybe have more even. Okay, so the second is in the category we touched. I think in the next decades, likely we will see that will be a wave of the transferring of the cell therapy from autologous therapy into the allogeneic cell therapy, which means it's off-the-shelf medicine now. Okay, the third one would be with the availability of more of those cancer therapy, agents. It can be checkpoint blockade, agent. It can be off-the-shelf cells. That we will see more of the combination therapy. Okay, and it probably will go beyond, just what we mentioned, and it can really combine with all the medical tools or strategies or treatment we have for cancer. So traditionally, what we have surgery, we have radiotherapy, we have chemotherapy, right? We have a precision treatment, which means those small molecule drug or antibody specifically targeting those particularly mutated, part of a particular type of cancer. And now we have this new things, immunotherapy, right? So the three categories we mentioned and beyond that. And all of those, all of those are available. So likely, for complex disease like cancer, it likely need more than one, one reagent, to treat it. So likely it's combination therapy, but it can be radio therapy within the category of cancer immunotherapy or beyond, right? It can be combined with surgery, combined with chemo, combined with radio and other precision treatment.
Of course. I mean, it's exciting to hear from your own perspective as we're looking towards the future, and we're excited to see the continued impact of cancer immunotherapies. As another question, I'm fascinated by your work, and I always love the side of being able to meld clinical research. I mean, with your own work, Lili, it's been amazing to see your work translate within the clinic. I'd be curious as we're looking towards the future. Do you have any thoughts or a look towards the future on maybe the potential changes or improvements in clinical trials in the future?
Yeah, certainly. So I think especially at my institution, UCLA, and in California, for instance, we now have this a really good support from it's a resource, called the California Institute of Regenerative Medicine. It really, supported my own work, many of the work from many of my colleagues, and all of this. And besides certain other, kind of foundations and at the private foundation, state resource, and the federal resources, we see in general an increase in investment and the industry side too into this cancer immunotherapy. Because again, the success, the very attractive future, like people view it, based on whatever achieved. So I guess with the more resources, again, go to the research side, go to the industry side, and this, nurturing, environment, like for startup companies to try out new ideas, we will see more of the new therapy being tested. And as a consequence, I'm a believer. I think some real, a kind of a working therapy will come out from that. You just need that base. You need to be tried, right? The idea. And there's always a gap between the preclinical study and the clinical study. I mean, human are human. There's no way you really predict what happened in human using any of the preclinical settings. That we do as good as we do. We keep evolving and using better and better model, maybe computational modeling, large data scale, everything. But still, the run out elected ideas still need to be tested out, and we need a base now, and we need a reasonable pace. The one thing hinder back, the development of the comprehensive therapy, like cell therapy, gene therapy in the past, it just take too long, to develop one therapy, and it's dragging years, usually over 10 years. At the time, the idea ready to be tested, and at the time, like even phase 1 clinical trial can be finished, it's just taking too long, and getting that get more resource, and maybe the regulatory, the FDA saw more and more of the gene therapy, cell therapy, and they're adapting, really quickly, to get from based on the pre-wealth experience, and more willing, to give permission, for the helpfully well-designed clinical trials permission, to happen. All of those are important changes. Hopefully, will continue to get better and really speed up the entire field.
Of course. Well, it's super exciting to see from your perspective, just looking towards the future of immunotherapies. And thank you once again for sharing. I just continue on this vein of translation. I want to pass it over to Minju to speak more on academic entrepreneurship and kick off our last topic here.
Yeah, thank you for sharing all your thoughts, Lili. So you've currently founded 2 biotech companies, Apia Bio and Immune Design, which was recently acquired by Merck. To highlight one, Apia Bio leverages stem cell technology and T cell development to scale these NK T cells for clinical approaches. So can you give us a little intro to the innovation behind Apia Bio, and in particular, the ACUA platform and the differentiation process from which stem cells turn into potent innate T cells, which are then engineered into these chimeric antigen receptors, or CAR T cells, to target cancer cells?
Certainly, yeah. So the Apia's core technology, Acra, platform, is based on the concept we touched a little bit earlier, at our conversation today. So the idea is we take blood-forming stem cells, and we culture that cells in a dish. Well, industry-wide, they are cultured in a bag, enclosed bag. That's easier to, get a large scale and sterile, and then it can be really expanded, like for the manufacturing line. We take those stem cells, we do the necessary engineer, but not over engineering, right? So remember, we talk about robustness and the simplicity and the practical, part of the translational approach. We do whatever absolutely needed, but not to overdo that. Okay, so we engineered the cell, we put the cell into the culture, and for a period of couple of weeks, the stem cell will be programmed and become this very powerful cancer-fighting cells. And because of the nature of the NKT cells we pick, this is a special type of the lymphocytes that can be freely transferred between people, don't cause problem. Okay, there's some deeper biology behind that, but it's very well, proved, and that's clear molecular base behind that. So now we have a large quantity of those cells. Like say, if we isolate the blood-forming cells from cord blood, one donor of the cord blood now can easily give rise to billions of the cells. And that can be formulated into the individual dose and then put onto shelf and ready, to use. okay? And the dose number based on the current successful CAR T-cell therapy dose, like we can, at the range from one, cord blood donor can make like 1,000 to 10,000 dose. So you do not need to make an individual one for each person now. You can make this once and it can be treated like 1,000 or 10,000 patient. So yeah, that is the platform behind that.
Yeah, that's a fascinating platform. So taking a step back, how do you think about crafting an entrepreneurial culture in your lab, in your own lab at UCLA?
Yeah, I think UCLA is doing a very good job, on this. When I myself get a little bit exposure, to the entrepreneurship, back during my PhD training at Caltech. As a graduate student, we were given the opportunity to take a class to learn about how to write patent and how to, kind of start a virtual, like, imaginary startup company. And I think those are very good exposures. Like, I myself still writing some most important part of my patent claims, I enjoy doing that. And knowing the language and knowing what happened at that part of the entrepreneurship, I think is kind of helped me frame my work, at the translational stage. When I joined UCLA, that I immediately get contacted by this technology development group at UCLA. So they are the entity, working for UCLA, handling all the IP related issues, matters. Okay, so the contacted me, I sit down with the officers, discuss about my research, so we figured out the translational value, of a particular project, and we think about the possible translational paths and maybe the possible, future like commercial opportunities over there. So it has been planned from the very beginning, and I keep a very tight contact with the office all the time, so we have the inventions happened, we file the patent protection, and we always coordinate, on the publication and everything. And the TGG office, also actively put me in contact with the from the business side, like the venture capital entities and other type of investors, and they're interested like in seeking for the new technology and the possible, commercial opportunities in the future. So all of those, so that's what I want to see is that for the two, at least for the two institutions, I have been experienced. Caltech for my PhD and post PhD training, and the UCLA for my faculty, career, that both have a quite good already existing structure to help the researchers give necessary training, but also help them, to understand the concept and help, step-wide, to get a invention, like from just the technology in the lab to a translational path, and eventually, in the case like the startup companies.
Okay, great. And as someone who's had success both in academia and now in industry, do you have any advice for young researchers seeking to translate their own work from an academic to an industry environment?
Sure, yeah. So I think it's just, one sentence of advice. Number one, don't be shy. And number two, don't be afraid. Yeah, don't be shy, because like traditionally, like the research, may consider themselves to be, we should be pure academics, right? We should just think about science, we teach a student, we do things, we test all those hypotheses and make discoveries on the bench. And something more than that is not something typically academic research should do. I think the culture is largely changed now. The new generations of the researchers probably won't really, hold this idea anymore. But in case that there's still some, remaining concerns, my advice is just don't be shy. Think about like what you discover on the bench do not have to remain on the bench and in the lab, right? If there's a possibility, it can really become something, help, patient, say cancer patient, why not? And you are the person making this invention, you know best of this technology, right? And you're probably the best person really can move it forward. And don't be shy, think about it and find the path and do it. The second is don't be afraid, is that, don't worry, like, well, I was trained, as a graduate student, I know how to do research, I know how to write paper, but I don't know anything beyond that. How do I do a translational work, right? So as I mentioned, many institutions, as at least the institutions I experienced, already have in place structure, probably now, even growing better and better, for many and even all the institutions. So understand that there's in place infrastructure and help and training opportunities, and actively seeking for that. Know about it, or just talk with people, or bring in the people to work with you who are the experts, right? And you, as a researcher, like my own strengths and my colleagues, maybe, the most strengths, we make discoveries. Okay, we should, stay focused, really maximize, what we're best at. But bring in the other people who are expert at the various stages. People can help you handle the patent, right? People can help you handle the interaction with the investment part, to move things forward. And of course, the clinical side, the clinician scientist, our own colleague or the other, people from elsewhere, they are interested, they can join and they can, help to, translate to the clinical side.
Yeah, I think this also lends really nicely into your teaching philosophy. So your research success and popularity is also reflected in the number of current students and postdocs who seek training in your lab, as well as many successful alumni that have come out of your research. How do you view mentorship in the lab, and how have you created a personal mentorship style for your students and trainees?
Yeah, so I guess that my experience is that I will give my students enough attention, but not overdo that. I'll give them enough attention, but also give them enough, freedom to explore on their own. It's a little bit fine line balance, sometimes hard to make, and almost for sure you need to craft it for individual students, because everybody is different. Some people like more, like they like to be nurtured more, at least at the beginning. Okay, some is pretty, kind of equipped and really want to, give more room for themselves to explore. Okay, so like for my trainees, my students, I try to assess a person, try to craft a plan, like an individual plan for them.
Yeah, as like a PhD student myself, I really appreciate how much thought it seems like you've given this to all your trainees. So you're also part of a small but growing group of powerful female scientists and co-founders for companies. So what can we do to further support and mentor this next generation of rising female scientists?
Yeah, so happy to, talk more on that. I myself, as a female, a scientist and academic, kind of members, we do see like a gradual loss of the female, like a portion, from the school moving up and to the more specialized career. The general understanding or belief is that the earlier stage of the career development, like one can receive for female, right? If one can receive guidance and support, and that will greatly shape like their decision for career paths in the future. Yeah, so in my own lab, we have like a different levels of the trainees involved, and each level contain, almost half at least, is female. So that includes the scientist, postdoc, graduate student, undergraduate student, and we sometimes post, high school student for some research. And I think like just giving this opportunity platform for female trainees, let them understand, they can see that the different levels, kind of people can move up, and that is indeed a career path over there. And you see this successful example, and you are ensured you can do it, and that's indeed a path there, for you to follow, to go up. And I hope like by doing this, like it will just open the door more for the next generation, female scientists. They give them confidence, point the path to them, and give them the platform they can mature and really kind of move up.
Great. Thank you so much for your time, Lili. I'm going to pass it back to Drew for some final closing thoughts and questions.
Thanks so much. Lili, just a few rapid fire questions to cap things off. As a pioneer in your own in immunotherapies, in the inspiring work you've talked about through mentorship and the excuse me, the path you carved through your own way, I'd just be curious to ask, do you have any advice for career, for early stage career individuals? Really hoping to pioneer new fields, get interested in the frontier?
Yeah, sure. Yeah. So I myself really like something said by Janus Salk, the person really invent, like produce the first polio vaccine. Some brief thing, very like courageous thing that's done in the past. He said one thing in there's one quote from him. I use it as my own motto, and then it's also for my lab. It's posted on our lab website. Over there, I want all my students to see it and get inspired as we do. Okay, so I can cite that for you. So he said, hope lies in dreams, in imagination, and in the courage of those who dare to make dreams into reality. I think this is something I would like to see, for the young generation, like have the good idea, creative idea, and don't limit yourself. Think big, dream big, and work. You need the work behind it to make those dreams into reality. But to me, that it's that being a scientist really gives us opportunity to explore new things, make discoveries, and then we need to, have the courage to dream and make that dream into reality.
Incredibly well said, Lili, and super inspiring as we're looking towards the future of new pioneers coming up. And as we are closing up the episode, I'd just be really curious. We typically love asking questions around the future of biotech, and one question we asked in the past is biotech in 2050. What is going to be the frontier in the next couple decades to come? As an immunotherapy pioneer yourself, I'd be really curious, how do you really view the future of patient care being impacted by cancer immunotherapies? Where are we in 2050?
Yeah, Drew, that's a very, good question. It's go beyond the 1 decade we just talked about. Technology is accelerating. It's true for the computer side. It's true for the artificial intelligence side. It's true, for the biomedicine, side. As mentioned, the immunotherapy really being, booming and show that it's working. It's in the past just about 1 decade. Imagine what will happen, kind of in another, 2 decades, 3 decades, 4 decades. I think it will be I would really, expect to see that the cell therapy, gene therapy probably will become really a standard, medicine and pill, like for many types of cancer by that time. And for the clinical practice and for the diagnosis, all those artificial intelligence probably can help quite a lot. Large database, there are many things probably can be done, through the patient and it's visual, a vision, they can self-diagnose, kind of do many things, or home test kit, all of those. It can also greatly reduce the burden on site at the hospital site. Okay, and with better and the more, broadly applied diagnosis that may also allow us to find the disease, identify the disease at the very early stage, that always, open up the opportunity for treatment greatly, right? And then with all those new, types of the therapies, right? Cell therapy, gene therapy, immunotherapy, and the choice will be more, right? There will be, better treatment. There will be treatment with less, side effect, and it will really kind of suppress or cure a disease or suppress the disease development, and meanwhile greatly improve the life quality. So I would say 2015, we will see a big change, probably on all of this.
Incredibly exciting. Thank you once again for joining us, Lili. As we're wrapping up here, any closing thoughts or shameless plugs? How can our audience hear more about your work?
I'm not really actively, on the social media, but I do have a lab website, maintained, liliyang.com. So we have all our new discoveries, publications, and activities, and the translation and the commercialization type of the activities, all posted on the website. So people would like to dig, a little bit deeper, that they can always come to visit that.
Super exciting. Thank you once again for joining us, Lili. We were super excited to have you on, and hope our audience enjoyed as much as I know Minju and I did. And just very grateful for your time. Thank you again.
Thank you, Jules. Thank you, Minju. Yeah, it's a great pleasure, to be here.